Hello and welcome back to the NWR Virtual Healthcare Conference. My name's Matt Wright. Next up we have Chimeric Therapeutics, which is listed under the ASX ticker CHM. And joining us from Chimeric is the CEO, Jennifer Chow. Before we get started, another reminder, if anyone has any questions, please feel free to type them in using the Q&A function within Zoom. Jen, I'll now hand it over to you. Please go ahead.
Thank you so much, Matt, and thanks for inviting me to be part of this year's conference again. I also appreciate everybody taking the time to let me introduce you to Chimeric, or for those that know us, to allow me to update you on our progress and our developments. So just pause briefly for our disclaimer, and then I'll go right into it. Chimeric Therapeutics is a clinical stage cell therapy company, and we have already had a lot of incredible achievements and milestones. We have positive phase 1 signals in glioblastoma with CHM 1101, or our chlorotoxin CAR-T, positive phase 1 signals in acute myeloid leukemia with CHM 0201, our NK cell platform. We've got 2 ongoing phase 1 clinical trials, and in the next 12 to 24 months, we have very meaningful and important clinical milestones to look forward to. We bring all this together with an industry-leading team of cell therapy experts that have proven experience taking assets from early stage development through to launch. So, Chimeric Therapeutics as a clinical stage cell therapy company. Why cell therapy and why oncology? For us and for my entire team, it's really about what we believe these therapies can actually do for patients. When you look at the evolution of cancer therapies, back in the 40s, we started off with chemotherapies, and chemotherapies were really indiscriminate therapies. The idea with a chemotherapy is it kind of bombs every cell. And so it can kill cancer cells, but it also kills a lot of healthy cells and makes people quite sick. In the 90s, we introduced targeted therapies, where our therapies were really, for the first time, going after a very specific marker on the type of cancer a patient had. And then in 2010, we were excited as we introduced immunotherapies, because immunotherapies were actually developed to stimulate a patient's own immune system to work against the cancer they had. Now, with each of the introductions of each of these assets or these types of therapies, we saw improvement in survival for patients. Chemotherapies brought improvements of survival from patients if they had no treatment. Targeted therapies then added on, particularly with improvements in shorter-term survival for patients. And then immunotherapies actually started to show long-term survival benefits for patients. So we've made progress over the course of the last 60, 70 years as we continue to develop cancer therapies. In 2017, the first CAR-T cell therapy was approved, Kymriah, and since that time, there's been 6 more CAR-T cell therapies approved in the United States. For the first time with CAR-T cell therapies, we're actually targeting or doing what both the targeted therapies and the immunotherapies do by targeting the very specific type of cancer a patient has, and also leveraging or activating a patient's own immune system to do the work. So what we saw with CAR-T cell therapies when we first introduced them was this incredible survival benefit for patients. And you sort of see it here on the graph, but probably where we see it most dramatically is in the images of patients that are 10 years out since their CAR-T cell therapy and remain completely cancer-free. And so what you see highlighted here are some of the patients that were treated with the early or very first CAR-T cell therapies in clinical trials. And last year, some of their outcomes, 10 years later, were published. And this has now allowed scientists to conclude for the first time that we can cure patients with leukemia, certain types of blood cancers, really with these cell therapies. So this is the incredible level of excitement that comes with cell therapy, because we really can, for the first time, talk about curative outcomes in oncology. Not surprisingly, when you're talking about being able to cure cancer, this is anticipated to be the fastest growing market segment in oncology. So from a market that's worth about $10 billion at the end of last year, we anticipate within the next 5 years, that market will grow to over $40 billion. So a very fast growing market segment in oncology. For Chimeric, when we really thought about this, we really want to come at this space very strategically. And so we built our company focused on 3 strategic pillars. First and foremost is having innovative assets. And so our assets are either first in class, meaning we are the very first people to have them and we will develop them and be able to commercialize them first, or best in class, meaning that they have features that are very unique. And all of our assets have very robust IP. The second pillar for us was about building the team that had the right level of expertise and experience in this space. This is a very specific and unique type of development space, and it does require people that have have experience and knowledge in cell therapies. And I'll highlight in a little bit how our team really lives up to this. The third pillar that we've really put in place for our company is following what we call commercially conscious development. And what this is all about is looking for the path that takes our assets from preclinical or very early stage development through to registration or commercial approval in the straightest line possible. And so really driving our assets forward to the patients that need them the most. We focus our development in areas of high unmet medical need, which allow us to streamline our development programs and in many cases move forward to registration pathways on phase 2 trials. And we've already seen, using this type of development plan, positive signals with multiple assets in multiple different disease areas. So when you look to develop in cell therapy or build a portfolio of innovative assets, there's different elements we can look at. We can look at alternative cell types, and you see highlighted on the left 2 of the most common types of cells that we use in cell therapy, T cells or NK cells. And then on the right, you see highlighted the alternative cell sources that are used in cell therapies. At the top, personalized or autologous cell therapies are when we take the blood from a patient and then we send it off to a manufacturing facility, engineer it, and send it back to that same patient. So the most personalized therapy you can possibly have. And then underneath that, you see what we refer to as off-the-shelf or allogeneic cell therapies. And these are cell therapies that are derived by a patient or a healthy donor coming in and providing their blood to be engineered for patients around the world. So this is the different types of cell types and cell sources that can be used when you actually start to build a cell therapy portfolio and pipeline. For Chimeric, we very strategically have brought in assets from the leading US institutions that actually give us both the personalized therapies that use T cells, as well as off-the-shelf therapies that use NK cells. CHM 1101 is an asset, a chlorotoxin CAR-T that we licensed from City of Hope in California. CHM 2101 is a CDH17 CAR-T for gastrointestinal tumors that we brought in from the University of Pennsylvania. And both of these are autologous or personalized CAR-T cell therapies. On the right-hand side, you see our allogeneic or NK cell therapies. And this was a platform that we licensed from Case Western Reserve University in Ohio. And this is a foundational platform that we can then build additional technologies off. With this pipeline and this portfolio that we have now, we really have an industry-leading diversified portfolio. And you see that when we look and we compare our portfolio and our pipeline across the different types of different cell therapy companies in Australia and also in the United States. We have that diversification using the different cell types and the different cell sources. So let me take you through a little bit more of each of our assets and where our development is today. I'll start with CHM 1101, and CHM 1101 is our chlorotoxin CAR-T cell therapy from City of Hope in Los Angeles. CHM 1101 was designed very specifically for patients with glioblastoma, which is most common but very lethal form of brain cancer. And certainly anybody that has ever known, a patient with glioblastoma can attest to how devastating this disease is. At diagnosis, patients are expected to survive only 12 to 15 months. And really what you see is once disease recurs for patients, their life survival or their life expectation is reduced significantly to about 6 months, very sadly. This particular CAR T cell therapy was designed for efficacy in glioblastoma. So it uses chlorotoxin to actually find and bind to the glioblastoma cancer cells. And it actually has an incredible story that chlorotoxin has been used as a tumor paint historically. It's been actually put into patients' brains, and what it does is it highlights for surgeons as a tumor paint where all the cancer cells are. So if you combine that with the cytotoxicity of a CAR T cell therapy, it makes perfect sense. You've got something that can actually find all the cancer cells and then send the signal to kill the cells that it finds. So really designed for that efficacy in this disease space. This is currently in a phase 1 clinical trial at City of Hope Hospital in Los Angeles. The trial you see highlighted here, it's 4 different dose levels. We started the trial at a very low dose of 44 million cells, up to 440 million cells, so 10 times the dose that we started at. The first 3 dose levels have been completed, and we recently announced that the first patient in dose level 4 has now been dosed as well. So starting to really move forward in progress and coming towards the end of this clinical trial. What we have available to date is data on the first 2 dose levels. So the dose level where we used 44 million cells and the dose level where we used 88 million cells. So the first 2 dose levels at the low doses. What we saw, and you see the data highlighted here, is that 5 out of the 7 patients across those 2 dose levels actually achieved disease control with their glioblastoma. Their disease stabilized, it did not progress any longer, and we've seen that now held for over 3.5 months with no dose-limiting toxicities whatsoever. So this has been very encouraging early in initial data, but very encouraging for us to be able to see this type of disease stability achieved in these patients with very aggressive disease. We're now moving forward and looking forward to being able to finish off this trial. So over the course of the next 18 months, what are we looking at? Well, in the first half of this year, we've already been able to complete dose level 3, start the first patient on dose level 4, and we're now initiating this as a multi-site trial to be able to complete the trial and prepare ourselves for the next stage. By the end of this year, we feel that we'll be able to show completion of the trial up and including dose level 4 and actually provide the preliminary clinical data for the trial before the end of this year. So really looking forward to that major milestone for us before the end of 2023. In the first half of 2024, we'll be able to provide final data because obviously there's some time that goes by before you're able to provide final data. And then we actually, assuming that that data is as positive as we hope it will be, and based on the encouraging signals we've seen to date, plan to initiate and move rapidly into a phase 1b clinical trial to continue the development of this asset. So lots of excitement over the course of the next sort of 9 to 12 months with this asset, completing the trial and looking at all of that initial data, and then moving forward into the next trial with it. I'll turn now to talk about CHM-0201. CHM-0201 is the allogeneic or off-the-shelf NK cell platform that we brought in from the Case Western Reserve University in Ohio. So this is a best-in-class NK cell platform. And by a platform technology, what I mean is it can be used to build multiple different assets off it. In our own pipeline, we already have plans to build 3 or 4 assets, but this certainly has the ability as platform technology to continue to build more assets off it. And we have partnership and ongoing collaboration discussions with different groups to really explore those opportunities. This particular platform technology, or NK cells, also can be used across a broad range of cancer types. So both blood cancers like acute myeloid leukemia, but also solid tumor cancers like colorectal cancer. And we've already seen, again, encouraging phase 1a clinical trial results. With this particular asset, we've already completed the phase 1a clinical trial. And you see highlighted here the trial design. This trial actually explored 3 different dose levels, starting at 10 million cells up to 50 million cells. Infusions were given to patients on day 0, and then 2 weeks later, a second infusion was given. And then patients were assessed for whether or not they were responding to the therapy. This data was actually published last year, and we were very excited to see, both from a safety perspective, but also from an efficacy perspective, that the data actually met all of the key endpoints. With a phase 1 clinical trial, first and foremost, we're always interested in the safety. And here, importantly, we were able to establish safety, and we were able to show that you can give cells from a healthy donor to a patient without causing any negative response, or what we refer to as graft versus host disease. So that was key and very important to establish. We were also able to show that these cells persisted. And really what that means is that the cells need to stay alive in a patient's body to be able to do their job. Here we were able to demonstrate in this trial that the cells persisted for more than 28 days. So they were staying alive to do what we needed them to do. And importantly, with NK cells, you want to know that they can expand so that you can actually manufacture from one healthy donor enough cells to really be able to support an entire clinical trial. And again, that's what we were able to show with this trial. When we turn to the efficacy side of the phase 1A clinical trial with the NK asset, what we saw was both efficacy in solid tumors and in blood cancers. In solid tumors, these were really colorectal cancer patients that had exhausted all other options for therapy and had multiple metastatic sites. 1/3 of the patients actually were able to achieve disease control. And within that, number of patients went on to have a durable response that lasted past day 100. So we really have seen this as a very promising efficacy signal. We'd like to see that disease control boosted higher into complete or partial responses, but this is certainly what we felt was very promising and encouraging in colorectal cancer. In blood cancers, we actually saw an even stronger efficacy signal, where we saw 100% disease control rate in acute myeloid leukemia patients. And noted there is an ongoing complete response. And I'll just highlight this particular case for people that are not familiar with it. This certainly has caught the eye of a lot of scientific attention, in that what we've seen in the NK cells in this first trial is a patient who has now been cancer-free for more than 2 years with the NK cells. So this particular patient had acute myeloid leukemia, had tried all other therapies, and came into the phase 1A NK trial. The patient received the cells as planned on day 0, and then another infusion 14 days later. By day 28, the patient's disease had completely stabilized, so it wasn't progressing at all anymore. And then, very excitingly, by day 100, the patient's response had actually deepened to a complete response, which means that all of the tumor cells were eradicated. We now know, in follow-up in discussions with the clinician that treated this patient at Case Western, that this particular patient remains cancer-free now more than 2 years since she received those NK cells. So a really exciting outcome for us. With the encouraging outcomes that we saw in this phase 1A clinical trial, our goal was really to move as quickly as we could to leverage that and move into either a combination trial where we could further enhance the efficacy signal that we were seeing, and also into engineering or further developing the cells. What you see highlighted here is the next trial, or the combination trial we were able to open up at the end of last year. This is a combination trial where CHM0201, our NK cells, are being combined with an agent called vactisertib. And vactisertib is a TGF-beta receptor inhibitor that actually modulates the tumor microenvironment. So what that really means is vactisertib's job is to make it so that the environment that the cancer is in is more amenable to the NK cells. This particular trial was initiated at the end of 2022, and we were pleased to be able to announce in January of this year that the first patient had been dosed, and now the trial is going through and enrolling additional patients. So lots of excitement around the early outcomes we saw in the phase 1A trial, and now moving into this important combination trial where we really are anticipating that we'll see even deeper responses from patients. As we look forward to what the next 18 months will bring us here with our NK cells, what we're looking for is having enrollment completed of all of the patients on this trial by the end of this year. And in the first half of next year, being able to look at that preliminary data, because you really don't start looking until all patients have completed more than 100 days of follow-up. So again, with the NK cell platform, very similar to 1101, we have an exciting set of clinical milestones coming over the course of the next 18 months. Now I'll just turn to our the third part, or our last sort of leg of our pipeline, which is CHM 2101. So this again is a CAR T cell therapy that is very personalized. And this particular CAR T cell therapy was designed for gastrointestinal tumors and neuroendocrine tumors by the team at the University of Pennsylvania. So it targets something called CDH17, and CDH17 is broadly expressed in gastrointestinal tumors, things like colorectal cancer or gastric cancer. And patients that have those cancers in the metastatic setting, sadly, again, less than 15% of them are surviving more than 5 years. The team at the University of Pennsylvania has been working on optimizing this asset for nearly a decade, and so we're incredibly excited alongside us to be able to move this towards clinic. And you can see some of the ways where they've really tried to optimize and make sure that we had the best CAR T that they could possibly develop and design to take forward to the clinic. The work that the team at the University of Pennsylvania did in the preclinical setting was highlighted last year in Nature Cancer, very prestigious publication, and it actually was the cover story. I think what really brought the scientific community to be interested in this and really generate that level of exposure in Nature Cancer was the efficacy that was seen with this CAR T cell therapy in the preclinical models. What we saw was complete eradication of all tumor in 7 different types of cancers. So over and over again, in all 7 cancers, what we saw was that all of the cancer cells were eradicated and there was absolutely no relapse. And that came with safety. And so we also saw no on-target off-tumor toxicity. So very important preclinical work that was highlighted and certainly has led to an incredible level of enthusiasm and excitement about seeing what this will do as we head this towards clinic. Over the course of the last 3 to 4 weeks, we've made a couple important announcements about our progress about moving this one towards clinic. The first announcement that we made was towards the beginning of March, I think March 9th, where we announced that we had completed viral vector manufacturing that would actually support the initiation of the trial. So viral vector is really the backbone of manufacturing CAR T cells, and viral vector can be delayed for many companies a year, 18 months to manufacture. So this was a really important milestone for us to have our viral vector complete manufacturing and be released. Just this morning, what we were able to also announce was that we've had a pre-IND meeting with the FDA and received very positive feedback. And so we're very encouraged by the interaction that we've had with the FDA, the agency, as it has been very much supportive of the planned clinical development and technical operation strategy for moving this asset into clinic. So very excited about moving this now forward to clinic. So across our pipeline, not only do we have the best in cell therapy innovation, but we also really believe that we focused on addressing high unmet medical needs in both solid tumors and in blood cancers or hematological malignancies. And you see highlighted here some of the disease areas that we're developing our assets across, and how much the patients in these disease areas really need new therapies to improve their survival rates. And then alongside that, you see the commercial opportunities that go with these different types of cancer. And so very broad, significant commercial opportunities alongside the high unmet medical needs that we're looking to address as we move forward. One of the things that I started talking about at the beginning when I was talking about the pillars for how we've developed our company is really about the experience and expertise. These therapies, as they're either personally manufactured or they're off the shelf, require very significant experience and expertise significant to cell therapy development. The team that we've brought together is small, but has extensive experience in the space, working for many of the top cell therapy companies in the world. And probably the most important piece to note is that there are currently 6 CAR T cell therapies approved in the US, and we've actually worked on 4 of those 6 therapies. So we really have the experience of taking assets from those very early stages through to commercial approval and getting them out to the patients that need them. Alongside us, we're very lucky and fortunate to have world-renowned scientists and clinicians as our scientific advisors. And you see highlighted our group of advisors that we work with very closely here. Dr. Lin, Dr. Bishop, Dr. Smith, and Dr. Maloney represent many of the leading cell therapy institutions in the United States and have really been instrumental in the approval of the initial set of CAR T cell therapies. And we feel very fortunate to have them working with us to give us feedback and their guidance as we continue developing our assets. So as we look forward to the next 12 to 24 months, we've set ourselves out a number of milestones, and you can see really here, and as I've gone through the presentation and talked about them, very much clinically focused. We've already, in the first three months of the year, achieved a number of milestones that were really important to us, moving forward with our chlorotoxin trial, manufacturing release of our CHM 2101 vector, and that pre-IND meeting. As we continue to move forward into the end of this year and through to next year, we're really looking forward to seeing the additional or the final data for our chlorotoxin clinical trial, and then also the data for the NK cell trial in combination with factisertib and moving our CDH17 asset into clinic. So you can see that highlighted here. So maybe just in closing, I will summarize and just, bring together, I think, what hopefully I've been able to show you across the last 20, 25 minutes. We really have a very innovative and diversified pipeline with first and best-in-class assets in cell therapy. We've already had very positive initial clinical signals, both in solid tumors with glioblastoma, as well as in blood cancers and acute myeloid leukemia. We're very much focused on optimizing our development plans so we can really advance our candidates from early stages through to registration as quickly as possible to get them to the patients that need them. And we have near-term catalysts for our assets in terms of major clinical milestones over the course of the next 12 to 24 months. We're lucky to work with world-renowned partners, collaborators, and scientists, and we really have developed the team that has the experience and the expertise to bring these patients through development to reach the patients that need them the most. So with that, I thank you very much for your time and your attention, and Matt for the opportunity to join the conference this year. And certainly, Matt, welcome any questions if we have time left.
Thanks, Jen. Yeah, we've got time for a couple of quick ones, so I'll just throw them at you in quick fire manner. So what's been the criteria and process for yourself and the board and management in selecting these assets?
Yeah, I think it's a great question. It's multifaceted. And so I think when we look at selecting these assets, we've looked at a number of things. We've looked at potentially what we believe in terms of their scientific promise. So we've certainly looked at how are the assets constructed, what does the construct looks like, what different elements are in the development of the construct. We've looked then at where they're coming from and the scientific rigor behind the group that's developing them and their experience. We've looked at then trying to look at what patient population they're applicable to. So are they applicable to broad patient populations? Are they applicable to small patient populations? What are the unmet needs of those patient populations? Is it a patient population that needs new therapies or not? We've looked at things like the IP that surrounds them. And then we've certainly gone and we've gotten external feedback from a number of scientific advisors around them. So I think I can sort of give you a sense of, in looking at any new asset, easily it probably takes 6 to 9 months from the day you sort of identify the asset to go through the process of really doing deep diligence to understand the potential the asset has, both from a scientific perspective, from a patient perspective, and also from a commercial perspective before we look at bringing them in.
And we've got time for one more. For the core NK vactosertib trial, should that be successful, what are the potential next steps?
Yeah, that's a very exciting trial, Matt. I think the core NK, the 0201 trial that is in place right now in combination with vactosertib. And with that particular trial, it's a phase 1b trial, so it's not a dose escalation. We've got one dose in that trial that's being used. If we see a signal in that trial, that we believe is strong enough to move on, and we're really optimistic that we will with that combination, we would then start to develop and have conversations with the FDA and the agency about how do we move into expanding the trial. So we probably, go from having 9 to 12 patients into probably somewhere between 24 and 36 patients. And how do we then move into a phase 2 trial that would ultimately lead to the commercial approval as a registration trial? So things can pick up really quickly once we see that initial signal off that trial, Matt.
Great, thanks, Jen. As I mentioned, we'll leave it at that. Thanks for your time today, Jen, and for the presentation. And that does wrap up the day for the NWR Virtual Healthcare Conference. We're back for day 2 tomorrow from 8:30 a.m. Australian Eastern Daylight Savings Time. So please, if you're interested in seeing some more of these sort of stories, just visit the website and you can register for further sessions there. But thanks to everyone for joining today, and thanks again to you, Jen.
Thanks, Matt.